let's keep the focus on science, not the politics. You are the future. So, anyway, summary, by the time we finish, the storm will be passed away. So it's good to be in the shelter of topics. So continue to keep it positive. So today we are looking into non-coding RNA and how this impacts for the science beyond just in the lab, but how now it can be go into the industry and also into clinics. So I will talk three things. First is about circular RNA. I believe that you guys just have a class to discuss a paper about that. And then microRNAs and how this turns into RNA therapeutics and diagnostics. So first about circular RNA. So circular RNA is a type of alternative splicing. As you know, in the mammalian genes, the exon and there is an interspace of introns that they are not contiguous genes. We have the genome sequence, and then when we transcribe it into a pre-mRNA, they will be spliced into individual parts and then come back together to form a mature mRNA. But then, because of this also provides a lot of opportunity to make different type of isoforms, like different variety, form the same genes, and this will encode like even there's 21,000 genes, coding genes. You can now even make more from this types. So a typical gene here, that is exon, and then the pink one will be intron. And if all exon is used, so you can see that this will be 1, 2, 3 exon. And then you can also do alternative splicing. In this case, there will be a skip. But from the exon 2, now it's just exon 1 joined to exon 3. And then together with the polytail, provided this is in frame, then you can make another type of protein. And then more than 90% of the human genes actually go through alternative splicing. But then today we're going to talk a special form of alternative splicing, and this generates circular RNA. So in this case, you can see that it's the same cartoon that I showed you earlier, but this time I show you 6 exons, and then in between the intron is in blue. Of course, you can splice all the intron out, and then you form this linear RNA containing all the exons. But now we are talking about a special type, that's exon 3, 4, 5, and there is some introns here. Usually they're base pair, so they bring the exon 3 and 5 in close proximity. In this case, when it's spliced, they will form exon 3, 4, 5 to form a circular RNA. Any questions? Yes?
Is it possible to move the zoom bar at the top?
Great. So, do you know what could make this type of base pairing? What kind of sequence inside the gene that could do that? Huh?
Hydrogen bond.
Hydrogen bond. Sure, that's they kind of have the base pairing that they can. But then what type of sequence is encoded in the genome that facilitate this type when they transcribe into it and then allow base pair? Yeah?
Just, I guess, like a CG-rich region?
It could be. So, but then it needs to be like, it's not just one sequence, right? It needs to be anti-parallel. So what could be possible? That there will be a lot of anti-parallel sequence.
Palindrome.
Huh?
Palindrome.
Palindrome, that could be the case. Yeah. But then here, there will be a sequence. What could be possible that this sequence will be similar to here? Yeah.
Should it be like a transposable element?
Yes, some type of repeat transposon element. So they will go this way, and then the other go the other way, and then they will be repeats, and that's usually help it to kind of basically.
Does that primarily happen within the introns to bring the exons into?
Usually it's the intron that encode, because the exon they may encode protein sequence will be less likely, but then the intron, the allo repeats, some of those would be corresponding. So this is the paper that I bring it to your attention. That is actually from Hopkins, one of the reasons. And I think this is a very interesting paper because when they were studying, as you know, they were just studying a cancer gene. And then when they do the experiment, can everyone see it? So, do you guys remember that paper? What did they do? How did they make this discovery? When they do a PCR reaction? Or have you read it? You guys discussed the paper last Friday or coming Friday? Oh, I see. So then this is a preview. So when you do a PCR reaction to know that it is occur for a splicing occur, you would put a primer onto the sequence. So in this case, if we want to know whether there is a transcript made between this exon 1, exon 2, and exon 3, for example, what you would do is to do a PCR primer, a forward primer on exon 1, and then another on exon 3. So when you do the PCR reaction, you will see okay, in the mRNA after they reverse transcribe to complementary DNA, you will see that, okay, this contains the transcript. But then what I like about this paper is that they do control. So what they did is that they also put primer here and then here. And then if there is, now we learned the diagram of the circular RNA. Say if we have this, E1, E2, E3. How would you detect a circular RNA? Which primer set will give you a signal? Yeah?
The second set that you're talking about.
Second set where?
Like this one, that one will,
So
E1
this-
will wrap back around to E3.
Yes. So that's how they do yes, and then you want to comment because you also raised.
Oh, no, I was just going like
Doing
this.
this. Okay, yeah, exactly. So because they do a control and they see a signal, they were so surprised. And therefore, at the end of the paper, they proposed a model. Maybe it's possible that there is a non-contiguous exon joining together. So that's the beginning of the circular RNA. But not much people really think about it until later on. And the principle that Hopkins is quite amazingly true after now in year 2000, that people start to find that's their joint accuracy at the consensus spike site, and then but they are not in the same order of the primary transcript. And they were found in normal and cancer cells, and in mouse and human origin, and then they have no poly-A. So this is kind of interesting whole truth. And this was discovered more when we have the sequencing technology. When people try to sequence, they find that, how come there is some transcript that is not as what you expect in terms of the order. Now this is quite an old review, 2014, but at least there are 25,000 circular RNA. So we were just talking about 21,000 protein for aging, but even for circular RNA, So I want to highlight enzyme here. This RNase out. What they did is that they will cleave the single-stranded RNA. So if it is a circle, it will not cleave, and that's why it causes an enrichment of the signal complex. And then this circular RNA is not just in the mammals; they can find in throughout all the different organs. Any question at this point? So if you look into the circular RNA, just like the RNase out, it cannot cleave as a exonuclease. So they because if you think about linear, you have the both end, you can assume it. But then for the circular RNA, there's no way to start, and as a result, that they have protected from the degradation. So in this case, what would be the implication about the property of circular RNA? Yeah.
It should last longer.
Yes, they have more. They last longer. So because they cannot, at least one class of the enzyme are not available to degrade this type of RNA, and therefore they have a more long life. But it's exactly at this point. I was giving this lecture maybe, I don't want to say too old, certain years, a single-digit number, when I present this, and then one of your alum asked me, and later joined my lab, so how they going to degrade it? And then we discovered a pathway, and that is called structure-mediated RNA decay, so this Joe Fisher. We discovered that there is certain enzyme that is doing endonuclease to cleave it in the middle. And how they detect which one to degrade is based on the structure of the circular RNA. How many base pairing it is. If it's too many base pairing, then this will trigger the pathway. So we will discuss more about the function of circular RNA and the microRNA section. Any questions before I move on? No? Then let's talk about microRNA. So I think many of you know about the gene expression cascade. The DNA makes RNA makes protein. And where circular RNA is focused on the last step, RNA, how they convert it to protein, and it could be regulating two-way, affecting the translation. How many copies of the protein can be made from a single mRNA, or how long the mRNA can last. And there are 2,000 microRNA, and they regulated at least 60% of the protein coding gene and implicated in many diseases. But this, maybe before we go into more technical details about it, what is your guess of the average size of the mRNA? How many nucleotides? 10 nucleotides to show. mRNA, you need to, no, not microRNA, sorry. Yes, just mammalian mRNA. Think about what is the average size of the...
Around 1,000 bases.
1,000 bases, slightly longer. Need to get longer.
1,001.
Double the size, about 2,200. If you think about it, divided by 3, then it's how many amino acids? So, minus out of it, so that's what it's not. And then the microRNA actually is 110 size. So it's just very small piece. And we will talk about the discovery, how to go find it. And this discovery actually starts from very small observation from animal that you find in the garden, just the soil, some worm about it. So this worm as C. elegans, and this is the wild type. What does it look like? And then they find two mutants that they interact each other. One is a huge baby. So that when they look into the morphology inside, there still is the baby stage, but they look very big. But then the other is a precocious adult. But when they cross these two together, then they go back to the wild type. So these two geneticists, Victor Ambrose and Gary Rockfield, know that there is interaction. And they know that LIN14 is a protein coding gene. That's they already find out, okay, we have this gene, and we delete it, they know that this is protein. But then they always don't know what is the function of LIN4. So that's, they have been trying, if you look back to the original paper, they have done a lot of different experiments. They have tried to insert a gene onto the LIN4 gene. They try to insert in a way that they know that it is likely not a protein coding gene. But then they do a very elegant experiment in my site to show that this is really not protein coding. You know what they do if you try to insert a gene? How they can prove that this is not protein coding? Yeah?
I mean, it could be like a reporter type gene.
Yeah.
If it was like GFP, and you would see it as the protein.
Mm-hmm. Yeah, they can put it in, but then how does it affect the microRNA gene? You can see that the GFP may be like if you put some transcription there, promote it. How can you do that? Think about a gene. What is a property for that it will be translated? Do they need to be in a special order, or do they need to be special number in order to be a protein coding gene? Yeah?
Could you alter the first codon?
Yeah, they alter the codon. So they can add, if you think about codon, they always are free nucleotides. So if you put DNA onto it, if it's a multiple of 3, will it affect the codon? No, right? Because they just move along. But what they did is that to show that it's not protein coding, what do you think they did? Because you say it's good to move the first codon, move a little bit. How many nucleotides would you test that this is really not protein coding? Yeah?
Do you just insert or delete 1 or 2 and see, does that change the effect?
Exactly. That's what they did. They put into the sequence so that it shifts the frame for 1 codon and 2 codon, and it still does not affect the microRNA. And that's why they know that this is not a protein coding gene. So if you look at that original, that is quite easy. And because of this discovery, they got the Nobel Prize this year, as I think most of you know about it. And science, actually, they know that this is not a protein coding, but what does it encode? So they present a piece of data in kind of like data sharing hub, usually at 4 p.m. You have beers and food, and then people just talk about it. And they present a piece of data. They are very excited, Victor Ambrose. They show that, okay, this is a marker, just to know what is the size. And then here's the wild type, lane. And then this is the mutants of the LIN4, the microRNA mutant. And then they find, by mapping, they can put a sequence back into the LIN4 mutant, and then they see the rescue phenotype. But then if they put tRNA, a non-related gene, then it will not rescue. So then they were very excited that when they probe it, they see that a hybridization sequence from the construct that they put in. when they do a northern blots, is how you run the total RNA on a gel and then transfer to a membrane. And then you can put radioactive probe that is related to the rescue construct, complementary, and then they bind on it. And then they were so excited. See, we got the band. Now we know why we cannot find it. It is about 15 nucleotides. They were very excited to share the whole thing. But then when they show it, then someone at the back of the audience is saying, have you looked at the bottom? So, because everyone thinks the bottom is the degradation part, nobody cares. And they say, and that person say, see, in the wild type, you see a bag. In the mutant, it's absent. In the rescue, you get back. And not in the tRNA control. So that is the size of the microRNA. That's how they discover. And this is because of the sharing of the data. Because I know you guys just start of the PhD career, so I see that a lot of, like, something to instill in you is about being open to share your data within your community. It's a safe space, because sometimes you may not spot something, other may spot. And this, at the end, lead to the discovery of the microRNA. And this another story about the sharing of the data. How does it work, the microRNA? Because now they know it's a 20 nucleotide. They have the sequence. They sequence it. And then they talk to each other. Now it's a story between Victor Ambrose and Gary Rofkin. So they share their own sequence, because at that time it's not like now. Every sequence is on the database. At that time you really need to manually run a sequencing gel and then mark what are the sequence of the LIMF4, the microRNA, and the LIMF14. Most importantly, so this accounts from Victor Ambrose. Gary Rofkin labs had identified evolutionary conserved sequence in the 3' untranslated region of LIMF14, in a region of the mRNA responsible for the downregulation of the protein LIMF14. And we and Gary knew that this sequence could contain the elements through which LIMF4 acts. Gary shared his LIMF4 UTR sequence with us, and we sent the LIMF4 sequence to Gary. On precisely the same day in June 1992, Richter and Gary independently noticed the anti-sense complementarity between LIMF4 and LIMF14. Richter immediately called Gary. Each of them read the complementary sequence to the other over the phone, particularly in unison. That was a very happy share. So that's the discovery of microRNA and how it works. And again, it's by sharing. So what they see is that, oh, there are 7 locations in the LIMF14 where LIMF4, the microRNA, can bind. But they bind in a very interesting way. It's not like, oh, it's exactly the same. There is a bulge in the middle that they don't base pair. And this turned out to hold true for nearly all the microRNA, how it works. And that's the beginning of the field. So then they think, oh, this is just an odd thing about LIMF4, and that is 1993. They published 2 cell-to-cell papers. Then everyone said, oh, there is something strange about the work. It could be, right? There may be some specific mechanism. So, and then everyone thinks that, oh, that is just odd. But it turns out that in 2000, Gary found another microRNA. And this is called LAS7. And again, it's in C. elegans. They think this is just developmental timing. Again, people think that, oh, there's something that is just specific for C. elegans for the development. But it turns out, no, that's not true. It turns out to be conserved, and even to human, mouse, everything. And I remember at that time, like, it's very exciting in the field about the microRNA discovery. What Gary described is when they discovered LAS7, what they did again is calling every friend that they have. Send me your organism, the mRNA, total mRNA. Send it to us, let us probe it. And that's why we see all the different microRNA. So then they start to think, oh, could there be more microRNA? So here's Victor's account about what's happened. The conservation of Les7 means that an ancient common ancestor of animals from sea urchins to humans had a Les7 RNA of precisely the same sequence as our Les7 today. After reading the paper in autumn, year 2000, I had to set aside 10 minutes to stare out the window and reorganize my view of universe. And that's very dramatic personal statement. Now we knew that LIM4 and Les7 RNA were members of an evolutionary ancient class of regulatory molecules. So it was finally obvious that there must be other RNA like them yet to be discovered in animals. Rosalind, Lee, and I, so Rosalind is Victor's wife, who has been helping many of the project as the first author of the original paper. Immediately set out to identify new microRNA by sequencing cDNA library prepared from size fractionated C. elegans RNA, so about 22 nucleotides. And then also something is there is the genome sequence of C. elegans and C. brugesi, close but not similar, but not so close worm sequence to see whether there is hairpin forming sequence conserved between these organisms. In retrospect, we were terribly naive about potential competition in this efforts to find new microRNA. We thought nobody else was looking. As it turned out, we escaped from being spectacularly scooped only by a whisker. By August 2001, so this a year later, Rosalind and I were enjoying a period of blissfully ignorant success, with almost one new microRNA falling daily out of the few hundred clones we have manually sequenced and compared to the raw C. brugesi genome sequence that was released in late July. We have identified a dozen or so new C. elegans microRNA that were phylogenetically conserved, some of them such as near one, all the way to human. Then, on the afternoon of Monday, August 6th, I received an email from the editor of Science containing the extract of a manuscript from Tom Tuchel Labs, asking me to serve as a reference, as a referee. To our horror, the abstract of the paper were bursting with reports of the new microRNA from flies and humans. I had to inform Science that I was unable to review the Tuchel paper on account of a conflict of interest because we had a manuscript in preparation with similar findings. We quickly decided to try an end run. Tuesday morning, we sent a pre-submission inquiry to Cell, because where they published, describing our identification of numerous new phylogenetically conserved small RNA like LIM4 and Les7. The editor responded that afternoon with the opinion that our finding would probably not fly at Cell, as referee would want to see functional characterization of this small RNA. We went back to Science heads in hand. And on Tuesday afternoon, the Science editor graciously agreed that if we could submit our manuscript by Friday, it will be reviewed along with the Tuchel paper. And by the way, an editor added, there's also a third manuscript. There was a serious problem with our promise to get the manuscript to Science by that coming Friday. It was already Tuesday night, and there was no manuscript, not even a preliminary draft. We have been planning to start writing perhaps in the autumn after occurring an imagined whole story about new world microRNA. So for 60-odd hours straight, Rostany and I did last-minute experiment, drew up figures, and typed while standing up at the bench to avoid falling asleep. We managed to send the manuscript off to Science Friday afternoon. It was accepted along with 2 other papers, although all 3 referees noted that our initial manuscript was poorly written to the extreme. Little did they know that it was the product of 2.5 days of flip-flop frenzy. So that's the beginning of the microRNA world. That we see a lot of great microRNA like the 2000 microRNA, and this is from the feedback science paper from David Patel, Victor Ambrose, and Tom Mitchell. So moral of the story, again, be open, because I feel like that if people know about each other earlier, then it may not have so much competition because everyone is hiding a little bit, but this is always a balance in your future career, like how much to share and how much to like know with good friends, and then also be open about your mind in terms of what could be the size of the products you're looking for. It may be not what I experience. So microRNA can be found in many types of tissue and even in cell-specific. And this particular to be the case in neurons. There are lots of specific neurons, they're just an expression type of microRNA. And microRNA are essential in development. So here's data from zebrafish. So this is the just a contrast image from the light microscope. You can look at from the side view, and then this is from the top. Like if this the fish, you can look on the top, and they are stained with fluorescent dye. And the green dye just show where the nucleus is, the DNA, and then the red dye is to show where is the brain stem. So you can see that if you're missing one key enzyme here, which we'll talk a little bit later, called dicer, then there is the morphological change of the brain, and you can see that from the fluorescent staining, you can see that the whole brain stems go away. But you just need to express one microRNA, which is highly expressed during the developmental stage, called MIR430. Then it can be totally restored. So it's just one microRNA is sufficient. But if you put microRNA, because it's 22 nucleotide, you can synthesize it, you can inject into the embryo. But if you just change 2 nucleotides at a certain region, which we will talk about later, called seed region, you inject, you won't see it. So this shows the specificity of microRNA, how it functions. So this, of course, if you have dysregulation, then causing disease. And you can see here that there are many types of disease that is involved, and we can also, like you can see that there's affecting of triglycerides, in cancer, in diabetes, and also in virus infection. So before I move on, I will talk about the mechanism. Any questions? So microRNA gene is like a typical gene in mammalian. It's transcribed by RNA polymerase II. So you know there are 3 polymerase, RNA polymerase, one major for ribosomal RNA, RNA polymerase II for many protein coding gene, but then RNase 3 major for tRNA, etc. Also RNA. And so here you can see that it's just a typical transcript, and they can be one characteristic is that they will be folded into a hairpin structure. And then this is called primary microRNA. And they can be just stand alone as a gene or is part of the intron. So here is a case that is a microRNA present in intron. So here you can see that is the long transcript containing intronic sequence. And then this is the microRNA gene. You can see that it's an intron. You can splice it out, but they can fold it into a hairpin structure, and then they will continue to process. Just I will tell you later, the processing mechanism is exactly the same, and then it will form a mature microRNA. Then one may wonder why certain microRNA would like to embed in the intron. While you can make your own gene, what is the advantage if a microRNA is embedded in an intron? Yeah?
Might allow like sort of co-regulation with that gene expression. So if you need some complex system, you want when this is expressed, a microRNA to like coordinatedly change something else.
Exactly. So that's what happened, is that there is a host gene, you make the mRNA to say it's a cardiac gene. And then they will express certain type of protein, but then they at the same time encode the negative regulator, the microRNA. So you only express in certain period of time. After that, they will suppress the host gene. So very good. So the next step after you transcribe it, fold it into a hairpin structure, then we'll encounter the first processing enzyme recognizing the hairpin structure. They'll cleave the end first, and then remaining 3' 2 extra nucleotide overhead. So there are 2 extra nucleotide here. And then this particular hairpin structure will be recognized by an exporter to go from the nucleus to the cytoplasm. And then this will process by another enzyme, dicer, will form into a double-stranded structure. Again, on the 3' end, there is a 2' nucleotide overhead. And one of the strands would be selected to go into a microRNA protein complex, where the argonaute, which indicated in red here, is the key protein to hold. You can think of microRNA as the key. What happened? So then the argonaute is holding this microRNA, and then it will have 2 possible functions. One is that it will bind into mRNA and then cause translation inhibition. Very persistent. So this is what we talked about earlier. When you bind it here, they will changing the number of protein that can be made from the mRNA. So it will reduce the number. Say for example, originally an mRNA can make 10 protein, now it can make 4. And the other function that the microRNA do is to degrade the mRNA, so reduce the steady state of that particular target. And it's usually causing the deadenylation, remove the poly-A tail first, and then that's what trigger the activation. And this is mediated by, there are 4 human argonauts protein, argo 1 to 4, and all of them can do this job. So now we will look into more details because it requires a specific way of binding. So here is the microRNA on the top, and then here is the mRNA on the top, and then the microRNA at the bottom. So when you think about any RNA-based parent, always remember that it's anti-parallel. Means that one from the microRNA, the 5' to 3', and then the mRNA that side is 5' to 3'. And then what they require most, it is true that it's only requires a front part, and as a result, in the very beginning, there is a bulge in the middle. Those are not required. And it turns out that it's not only just this part is not required base complementarity, but also the 3' end. Only the 5' end of the microRNA is required. And this is from the 2nd nucleotide to the 7th nucleotide. So just 6. So how many possible microRNA with different sequences? Yeah?
Where do they stick?
Yes, 4 to the power of 6. So that's how much? Okay. So let's do an exercise together. I'll give you a couple of minutes. Say this is the microRNA 21 sequence. What would be the possible sequence on the mRNA that you can recognize? So I'll give you a couple of minutes. Think about the sequence on the mRNA, how it would recognize. Any questions on the exercise? You can write down, here's your sequence. What sequence on the mRNA it will recognize?
Yeah,
That's very nice. Is there water? You guys pick this.
Anybody have hand sanitizer?
I have a little bit.
I do actually. You guys want to try it?
I don't know if there's a like a thing in there.
We also have hand sanitizer if you
Okay. Let's see what this is. Thanks. Maybe it helps with the. Okay, any volunteer about the sequence? What would be the possible sequence for the mRNA from 5' to 3'? Yeah.
So that first segment, we've got, are there any of these markers?
Yeah.
So this would be our seed sequence,
Yes.
and so we would have the reverse complementary sequence on our mRNA if it's 5' to 3'.
Yeah, very good. So you basically focus on where the C sequence is, and then this one base pair to here, and then here, here, and then so forth. That's great. All get it? Oh, good. Good job. Thank you.
No problem.
Okay. So this is the sequence, UAAG. But there could be other possibilities as well. So how could that be possible? Isn't that you tell me just base pair?
Partial base pairing and third looping.
Huh?
Partial base pairing, third looping.
No, it needs to be exactly this. It needs to be base pair to the C. So it cannot be. This 6 is required. What do you mean by looping?
It doesn't have to be 1 to 1.
Oh, this need to be 1 to 1. Yeah.
The seed does, but the rest.
The C need to be that. Yes, yes. But then even this base pairing it could be slightly different. So maybe I don't know whether you guys encountered this yet. There is the Washington crate base pairing, but there is some additional one that you can base pair. For example, G sometimes can base pair with U instead of just C, and this open up like additional. As you can see, like here, G can. And there is other modified nucleotide like inosine that would also provide other possibilities. So this is the rules for the partially complementarity. Now we're talking about exactly complementarity. If it is exactly the same, it happens with one human microRNA, but it happens a lot in viruses. That's the microRNA from the viruses will bind exactly the same for the in the plants. Viruses, they will bind exactly the same. And in this case, it's called a cleavage. Instead of degradation, shortening the poly A tail, now they will cleave at the middle of the where the mRNA binding to the microRNA is. And I will show you a more detailed picture. And this is only possible with AGO2 among the 4 others. So here's the picture. So here's the microRNA at the bottom, and then again the top is the mRNA. Now, if you look at it, is between the 10 and the 11 nucleotide. So in this case, if you count like here, it will cleave at this position. So between 10 and 11 on the mRNA. Okay? So now I give you where would the microRNA 21 bind to this mRNA? They're exactly complement. Where would you cleave? Any volunteer? Maybe at the back. Any volunteer from the back? So counting exercise. Wanna try? Everyone at the back is kind of like looking very intentionally. I presume you guys can do it. No, anyone? Anyone in front as well? Willing to try?
It's fun.
Okay.
And this is really simple and that's what's really stupid. But just the 10th to 11th nucleotide is going to be right.
Yeah, from here.
From here. Oh, okay. Got it. I was like, I'm born.
microRNA.
On the microRNAs, we go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Right there.
Where it will click? Is it on the microRNA or the mRNA?
It'll click on that mRNA. Sorry, this position.
Yes.
On that mRNA.
Good. Very good. So this is it. Very good. Thank you. Thank you. Good job. Good. Okay. Any question about the regulation? Yes.
So is this cleavage also going to cut the microRNA?
Mostly not, but over time, people find that this could be a mechanism for microRNA degradation, but usually it's not, but over time. Other questions?
So it's sort of marking where the cleavage will be, but not actually performing the cleavage itself.
Yes, it will
Okay.
cleave the mRNA position. So they have the residue pointing to this position for the nucleophilic attack. Okay, good. So now we talk about more on the regulation. I will go back to the-
So if it's not a perfect match, it will not cut.
Not in this way. In the cleavage, it will. Okay, so because you know the microRNA from the genome, you can do a prediction because the rules is only 6 nucleotides as the key to recognize where is the mRNA target. So people have already done, like in early days, computationally, how many microRNA typically on mRNA. So that's average about 4. So I just give you some scenario what it could happen, because not all the microRNA could be present at the same time. So this is an example. That's an mRNA with 3 microRNA binding sites, 3 different binding sites for 3 different microRNA, A, B, and C. And we know that there is already existence of microRNA A and B present in the cells. So they are repressing it. But maybe in a condition that there is stress, then there is the expression of the microRNA C. And in the top case, then you can see that if there are more microRNA occupied on the mRNA 3' UTR, then this would causing the expression go down. But in another scenario, it could be like microRNA B suddenly go away in the stress. Then this mRNA expression will go up. So this could have implication later on how the regulation on the microRNA on the mRNA targets. And in terms of the microRNA, like we're talking about changing level, how could this be possible? One is could be changing the stability of the microRNA. And then the other is could be transcription, you make more, or at the processing step, like the Georgia and Dyson, that could affect it. And this is just an illustrated example on how concerted the effort during DNA damage. It can regulate the stability, it can regulate transcription, and then processing. And it turns out these microRNA, they all towards a function is to promote cell growth, arrest when there is DNA damage, and also apoptosis. So one interesting aspect people find that is that microRNA, when they look into their targets, when they group into what is the function of this target, they are under the same pathway. So this gives us an idea, like microRNA is not just regulate single gene, but multiple genes. And in a sense that it is like a master regulator to coordinate what change of a certain pathway. And this is an example on microRNA 34 during DNA damage, and P53 were induced, and they are all towards the growth arrest.
More like a transcription factor than
Yeah, some of them is transcription factor. So I will tell you later on, there is an example. By thinking about microRNA and then the transcription factor, how they can causing different type of feedback loop, and we will talk.
So when it talks about a MER family, does that mean that it contains the same seed, but everything else is just...
Good clarification. Yes, if it is same family of MER 34, it means that they have the same seed. The other sequence could be different. Other question? Yes.
What could be the significance of the same seed but different down, I guess, yeah.
Why, so your question, I rephrase it. So why there are so many, there could be different microRNA share the same sequence, but they may have other like sequence. Then the answer would be is that other sequences play a role. So that may be modulating what type of effect or the stability of the microRNA under certain developmental conditions. So one good study, one active researcher here studying that is Luciana in the engineering department. So she studied East Fluorinate conserved microRNA family. Other questions? So there could be different type of regulation. One type we call it a decoy. In this case, that you have all the microRNA the same. They can go to a certain mRNA target and suppress. But maybe in stress condition, you express some new mRNA. In this case, it's not microRNA change, but the mRNA. And they have the same sequence that they can match to regulate. So in this case, you have think that the expression of the new mRNA target causing a dilution of the same amount of microRNA. So in this case, the black transcript, now they have higher expression because you have the same amount of microRNA. And this happened actually for the circular RNA that we talked in the first section. There is a family of circular RNA called CIRS7. They have a lot of binding site from MIR7. When they express to it, you can think that they are sponge. They absorb the existing microRNA and regulate the pickup of sequences. So here's a more detailed example. So why do people know that this is a sponge? Again, this is looking into the brainstem. And then you can see that if you express a lot of this circular RNA, then this particular case, then you can see that they have a lack of development. But you add back a lot of microRNA 7 in the cells, then they restore. So here is a summary of what people have found. That in the normal cells that you have the hybrid size, this is what they are looking at it. It's normal, and then there's a lot of microRNA. And then the microRNA 7 target expression is low. But then now, if you express the circular RNA, because they have 70 to 80 microRNA 7 binding sites, they kind of absorb all the freely available microRNA 7. So there's little of the microRNA 7, and then the microRNA 7 target original expression will go up. And this we call a small hybrid. But you can now putting back with extra microRNA 7, and you restore to high level, then it resumes the microRNA 7 target. Yes.
So, are these circular, like, circular RNAs, are they basically invulnerable to like the AGO? Like when they're soaking up, can they just continually like have these miRNAs attached to them, or will they also be degraded through the miRNA target?
Oh, so when they bind, good question. So when they bind here, it didn't show, they are not the 22 nucleotide base pair, so it's not a cleavage effect. Instead, they are mainly just for the C that they bind. So it's more like stuck in there. Yeah. So it's not like causing the degradation. Other question? Good question.
So are the mirrors usually using? Because if there's too many, then this sponge thing.
Yeah, so it's limiting in the sense that when you will be sensitive to that, you can express it. And this actually is a method that researchers do. One way to reduce the amount of microRNA present is to express like a GFP construct with a lot of microRNA, specific microRNA binding site. Then you can deplete the internal food. And this turned out to be important. There is a correlation. People have shown that there is high amounts of the circular RNA in normal individuals, but for the Alzheimer's patient, there's less circular RNA. So there is a correlation. So the other that I want to discuss, apart from the targets could be expressing artificially or inside the cell suddenly induce certain type of microRNA target that will change the availability of microRNA protein complex to regulate. There's another way to regulate the microRNA is changing the mode. Because we talk about certain microRNA will cause translation, suppression. Some is by the mRNA degradation. They all have the argonautic protein, but this is dependent on what other protein is associated with the argonautic complex, will determine whether they go for the translation inhibition or mRNA degradation. So one way that people have found that during the stress, they can change the balance because it's the availability of one other protein complex with the argonaut, so you can modulate. So my question to you is, what is the implication in terms of the transcriptome and the protein for these two different types of regulation? Shift to, say, if we shift to the mRNA degradation more, what would be the implication to the transcriptome and the protein? mitochondria around, yes, if you go for that. But then say, what is the difference? If it is more majority for the mitochondria to work in mRNA degradation versus it's more majority working as a translational inhibition. What could be the difference? Yeah?
Translational inhibition can rapidly be turned back on, whereas mRNA degradation is gone and you have to reproduce it.
Yeah. So one is more irreversible. When you degrade the transcript, then, as you say, it takes time to build it back up. And while the translation inhibition is you stop just how many copy of protein. If the microRNA is like go away, then you can immediately go back. So one is more for the irreversible switch. And that happens particularly in the zebrafish that I showed you earlier about the microRNA 430. They are the developmental shifts that they suddenly trigger a lot of microRNA 430 target degradation. So it's driving the developmental state to another. So this is the difference between the two. Now is the time for any question before we go into a different scenario.
Are microRNAs only inducing?
What do you mean?
They only affect the cells that they're made in?
Good question. Majority, yes. But what people find that they can also export out in exosome, and then it is proposed that it can also absorb into other cells and then affect other cells like expression. The key would be how much. Because people do detect exosome, there is microRNA, but how much is sufficient to cause another cell? So this is the question that in the field hasn't proven that yet. But there is a possibility that that is possible. Okay. So we talked earlier that microRNA regulates on the transition from RNA to protein. So, and then transcription factor is affecting how many RNA being made from DNA. So what if these two work together? Then we have something to consider. About the half-life of the microRNA, they usually tend to be long. They can be days about it. That's once they are made. And the other is exactly the question that you just posed. For microRNA to work, it needs to pass certain threshold. It's too little, somehow maybe it's just the mechanics of the biochemistry. It's not sufficient to bind sufficient target. So you need to pass certain threshold to cause depression. So what I mean is that if there is an mRNA being expressed, that is a target for microRNA. MicroRNA also increase the expression, but it will not affect the mRNA expression until it passes certain threshold. So now I want to put a scenario like below. That you have a transcription factor going to make the target of the microRNA, where microRNA can suppress it. But the transcription factor is also making the microRNA target. So just like we talked earlier, this could be the case for the intron, right? You make the mRNA, but you also make the microRNA at the same time, co-regulation. So you will see that there is increasing expression of the mRNA because microRNA, although it's made, but it does not hit a threshold to inhibit yet. But then once it hits a threshold, then it will start the inhibition of the mRNA target. So this creates a pattern we call it a pulse. So you first made it, and then it goes low. So now, I'll give you some exercise time. Give you 5 minutes, so everyone has time. So here are different scenarios. I show you one example there with your house that you can create this. Biology is very interesting. They have a system. You can create a certain pattern of expression. So you'll see a gray dot here. This is the initial expression of the target. Okay? And then just look again.
Causes mRNA long half-life?
Sorry?
Why is mRNA not long?
Not long. Maybe the enzyme to degrade micron is not very active, but the other thing is that micron is about 22 nucleotides, and the content that bind to it really protect it from any degradation.
Oh, I have, I think this.
I don't think that's right. Sorry, I'm trying to rationalize these to myself. If you, I guess my question, how do you know if like the transcription factor or the microRNA has the nitrogen effect? Would you just
We don't-
assume it's the, oh, okay.
Yeah, we don't know. I mean, so there could be many possible
Yeah.
patterns, but then, yes.
Just the, okay, kind of the balance between the two.
Yes, yes, very good. Question?
I don't think so.
So there could be many shapes possible. We're just talking about the red is which. Okay, okay, anyone try this one? Open up the screen. Let's go on. That's good. Any suggestions? I think, can you explain like why you think it is way?
Yeah, because there's no microRNA to degrade the target, so it would increase pretty quickly and then eventually would reach like a steady state.
Okay, good. So because they already increased and then now it's even increased further because the lack of microRNA, and then now you get to the steady state. Very good. Okay. How about this one? Anyone? Yeah. Want to try?
I'm not sure. Reverse function.
Reverse. Okay. Please show me. And then. Thank you.
I might be wrong.
It's okay. Scientists need to learn how to be. Okay, why do you think that is the case?
There's time difference. First, increase target, and then inhibit, right?
Uh-huh.
But first, increase target, and second, like increase the inhibition of microRNA. I mean, decrease of inhibition of microRNA
Yeah.
means like going back.
Okay. Okay. Any other suggestions?
I think it would just stay low.
Yeah, it could be just very slow.
All right.
Yeah, because maybe there's not much target to be activated because it's suppressed. So it can just go down. Yeah. How about this one? There could be many different ones. Because say there could be more target. May not be going back to the very top, but I think likely to go down all the way. Yes. You are the most active task among the IFG. Yeah? Yeah. Very good. Very good job. This is the first time every answer has someone come up. Very good. Yeah. Okay. So why there is a change? I see that there is a change of slope. Why that is the case?
So first you have the transcription factor working, so that's the initial slope.
Uh-huh.
And you add in the effective mRNA after you reach the threshold, so it becomes like additive.
Very good. Very good. Okay. Very good. Thank you very much. Good job. Good job. Basically, what you guys do, I didn't put into the further, you guys can even further than me. Get to the steady state, I really like discuss. Very good. I will update my future. So you can see that this one will go up and then further up. And then this one will go down quickly and then slower because the microRNA is suppressed the effect, and this is a reinforced decrease. I think you guys will get the notes later on, don't worry. You guys will learn. Okay. Any other questions? There are also many other type of different circuitry. If we talk about, Elaine earlier mentioned about the transcription factor, imagine that the target is a transcription factor, and that transcription factor affects the microRNA. Then this would create a negative feedback loop. It's very fun. It's happening inside the cell. They are very clever how it was designed. So take a look. Okay, we'll end with.
I just have a question. There are a lot of papers talking about microRNA. When I'm reading literature, sometimes I'm like not quite sure how to evaluate the strength of the conclusion. I know that most of the microRNAs are not most, but like often they're not a perfect match. So you would not expect RNA to be by PCR.
Yeah.
But almost a lot of them in America are also.
Yes.
Yeah. But what's the average? What should we expect for a random microRNA?
I think most of the microRNA target you will see lighter degrees. So that is good. But then you want to ask about the strength of the data. Usually what you would do, what we would do is that clone the target, and then on the reported gene, and then you have the microRNA present, and then see whether it can repress, and then you mutate on the seed region. And if it does not, then this seems to be specific. So that's something to do. And then other things in our human genetic class, of course, look into whether there's mutation. And if there is, and there is a corresponding change of the target, that would be another very good sign about whether it's true. And then the third thing is look into the conservation of the target. Usually they conserve across. Other question? Okay, so we learn all these things. It's good, molecular biology, we learn all the process. But starting from the beginning of all the molecular biology discovery, is also have a very good application. Think about, we learn about DNA replication, we have PCR. That's now we can even detect very small amounts. For microRNA, it turns out to be a very good therapeutic and diagnostic tools for us. So I'm just going to show you a small video from a company talking about microRNA.
We'll also be discovering how their dysregulation leads to disease. Typically, when a microRNA is dysregulated, the pathways that are the targets of the specific microRNA go awry, which disrupts the delicate balance and harmony of normal cell function. MicroRNA medicines act by targeting dysregulated microRNAs, resulting in the normalization of genetic pathways and thus halting the disease process.
So of course it's just an advertisement. But then I think it speaks some truth about microRNA. If you think about drugs, usually we target one target at a time. But this will cause mutation. And just like even you have very good small molecule, if you have a family member got cancer, you sometimes heard the worst thing to hear is that there is resistance of this drug because they can go around it. But then microRNA, at least this is the promise, is that it can affect multiple targets under the same pathway. So even one gene mutated, the other may not. So this is what they have been looking into, is that microRNA affect multiple targets, and they target the pathway of the human disease. So there are multiple ways people have been doing it. One is called Athg. So basically means anti-microRNA. What they are chemical species like the RNA backbone. They can bind to the microRNA in a complementarity manner. So you can think that they dilute out the microRNA. And originally, the microRNA would be suppressing the mRNA target, reduce the protein production. Now you have the endomere, then the microRNA cannot function. So the expression restored. So Athg has been, as a therapeutics, or at least have been tested in many animal models. So what I showed you earlier, all these slides about like different disease, they are doing the same approach. They put the inhibition of microRNA 33 A or B, means that it share the same sequence. They can raise the high density lipoprotein or the very low density lipoprotein triglyceride. And then they can do other things as well. And then the other ways that people have been thinking is this is the way to reduce the mature microRNA. Then how, if we want like more microRNA to be made, what can you do? So one way that is, can you put, at least to test whether it functions, is to express the microRNA, because it's just a typical protogene. You can put in a viral construct, and then put microRNA to say 26A. People have done that, and then have shown that this is good for cancer therapy. What they did here is that they, this liver, the normal color is brown. When there is metastasis, they will be this pink cell. So what this study show is that if you express microRNA under inducible promoter called doxycycline, you add chemicals, then you can increase the expression. Either you just put a control versus that expressing microRNA 26. Then you can see that there is no longer metastasis. And then the third thing that people have been thinking is to use, because microRNA is just 20 or nucleotide, you can synthesize it. And when we did that, we call this small interfering RNA or siRNA. So you can chemical synthesize double-stranded form and then go into this pathway, go inside the cells, then they will perform like microRNA. So this is what people have been looking into, like the siRNA. There is the double overhang, because that's what looks like inside the cells how microRNA process, with 2 nucleotide extra at the end. Then we need to think about the C region. That is the 2nd to 6th. And then this will bind to the mRNA. But then there could be a problem. But then when we load into the cells, we can imagine that it can be on target. It's perfectly complementary. This will causing the siRNA cleavage of the mRNA where you want, which is great because imagine it's 4 to the power of 20 to find out a target. So this will be very specific. But just like we talked about earlier, this will be mediated through Argonauts too. You can find that target and then treat particular mRNA. But there could be a problem as well. Why that is the case? Because we really very specific. 4 to the power 20, put a specific sequence. It will be target this gene, will kill this particular mRNA. So low expression level, very good. We can be doing that. But what could be the problem? Also from the same pathway. So we're thinking about using this pathway. Put the siRNA in, load it. We know the sequence exactly complementary. Cleavage 10 to 11 nucleotide. What's the problem? What could be the problem? Yeah.
These target hundreds of mRNAs, so it can go to the partially complementary, or it can do a wobble effect and go hit some other ones.
So it's like it's true. We can define, but there could be the partial complementary effect, and that would be the off target. So this is the unintended target. You have the on target, but you have the off target because they just require 7 nucleotide, 6 nucleotide in total, and they will bind to those. Yes.
So if people are designing these, people created algorithms to basically look at all the 3' off-target effects from a given construct.
Yes. People look at it and then they also measure it to see whether that is possible. So that's the power of now we have all the genomics data to do that. But then there is also hope because they learn about the chemistry and the biochemistry of how Argonauts work. They can design a particular modification so that it won't go into the other pathway. And then it will be more on target to causing the siRNA. So, but this is like some super modification. So this has been using in many therapeutics, like microRNA, people have been looking into virus, cancer, and then siRNA to particular target, people also look into that. And this has been going up and down throughout my teaching here. That it's good, it's the cure of the world. And then the next day it will be like, oh, the whole stock market come down. And it's always down to off target, because it could be delivery problem, toxicity, because although like the people already identified the short RNA likely not trigger immune response, but in certain occasions it still causes something that we don't understand. That's immune response and also the. But to start with, there is already a first FDA approved RNAi drug. So this through the chemical synthesized siRNA pathway. And one smart thing this company did is that they target liver. Because all the drugs first go there, so there is no delivery problem. And they particularly target this TDR gene, and this would because if there is too much TDR, they will cause some amyloid fibril, and this would causing heart failure. So they will stop that. So at least there is something working at the moment for the siRNA. Okay, before I move to the end for the diagnostic, any question on the therapeutic about the 3 ways that you can modulate? No, then I will end with microRNA is also a very good biomarker. As you could imagine, because microRNA is a master regulator for many different genes, so the expression pattern could likely affect the mRNA pattern. Instead of studying 21,000 genes, how they express, maybe you look into the controller, then this would provide. And this is just one example, but it will say that 2/3 of the sample can classify cancer of the origin. So this is the piece of data. The blue signal here is saying there is a decreased amount of the microRNA, and then the red signal is saying decrease. And you can see that they can characterizing by their tissue origin. But one thing that this study also points out is that if you look into the normal versus tumor, what is the major pattern you saw? Instead of just focusing on, oh, this can segregate their tissue origin. Look at this the normal, and then this the tumor. What is the major change you see in the tumor case?
A decrease in expression?
Yes, it's a general decrease. Somehow tumor, one way is to take out the regulator. It's deregulated, then it may go awry, then it will be to that back. And that's what one of the observations. And the other things that, apart from using like a tissue sample, you measure the microRNA expression. People have been also looking into like circulating microRNA in the bloodstream. So we talked earlier, can it go to another cells? And that's the reason why people think it. Because you can find in the blood, there's different type of microRNA. Maybe this would be a good way for like further site regulation. But this also can be served as a biomarker. And I'm particularly inspired by this PNAS editorial say that are we bind by the potential? And people have been looking into it because into many different body fluid, you can see here there are many different types here. They also find microRNA for whatever reason. And people have been thinking, can we do this? Because sometimes even if you get a tissue, do a biopsy to take, it's very invasive. But if you can just take some from blood or from other body fluid, it will be right? And there's a few reasons why microRNA is a biomarker. They're very stable in fluid. So you ask the question, it could be because argonauts protein complex is quite big. They already protect most of the microRNA, so it will not allow degradation. And then people have tested that the fresh sample and the fixed sample is the same. So this again helps to be for the identification. Okay, that's all. I think we just make it a trip. If you have any questions, email me or come find me. Okay, good. Any question that I can answer at this point?